special interests are in the bone marrow transplantation, blood stem cell biology, and especially blood stem cell aging. And there is a lot of connection between the glucose metabolism, glycogen accumulation, and aging of hematopoietic stem cells. So today, the title of my talk is on glycogen accumulation, glucose metabolism, aging of human hematopoietic stem cells. And it is based on our previous comprehensive proteome studies of human bone marrow cells from 59 individual healthy subjects, human subjects, then between 20 to 60 years. And this study showed that one of the most prominent findings is an upregulation of proteins involved in insulin processing, glycolysis, and glycogenolysis, in addition to numerous changes that have been described in murine models of aging of hematopoietic stem cells. But this is for the first time done in human, and then a comprehensive proteomic studies throughout the ages between 20 to 60. And especially remarkable is that this upregulation of glycolytic enzymes in the upper half of the glycose metabolism is seen only in hematopoietic stem cells and not in the other populations that we have examined. These other cells are the lymphocytes, the monocytes, the granulocytic precursors, and also the mesenchymal stromal cells, as well as the erythroid progenitors. And as you see here, they are not, this upregulation of central carbon metabolism is only seen in the hematopoietic stem cells and not in all the others. So, yeah, and we subsequently demonstrated that this is caused by glycogen accumulation and elevated glucose metabolism in a subset of hematopoietic stem cells that has become more glycolytic and not on a per cell basis. So the fraction of glucose uptake high hematopoietic stem cells in a healthy subject younger than 30 years is almost minimal to non-detectable and maximally up to 1%. And whereas in elderly, in those over 60, for example, in this human subject over 70, we see up to a maximum of somewhat, in this case, 5.6 and up to a maximum of 8% in those over 70 years. And based on this, then we have been able to separate the hematopoietic stem cells from humans then into these three different categories, especially in the elderly. And to define the molecular characteristics of these hematopoietic stem cells with these three different levels, distinct levels of glucose metabolism, we have performed a single-cell RNA sequencing studies, single-cell transcriptome studies on these three subsets from altogether 6 healthy human subjects, approximately 700 single cells from each category from these 6 different individuals, and were analyzed from each of these categories, 700. And between 26,000 to 34,000 genes were identified from each single cell. And then for comparison and as a reference, we have also performed transcriptome profiling studies comparing CD34, the whole CD34 population derived from old versus then young subjects, and gene ontogeny comparisons, as well as multi-layer gene set enrichment analysis studies were then performed. So the glucose uptake high subset is characterized by upregulation of genes involved, especially in cell cycle arrest and in inflammatory and stress response, inflammatory and stress response here, and also in apoptosis and survival pathways, upregulation of apoptosis and survival, other survival pathways. And this is the comparison between the glucose uptake high versus glucose uptake low single cells. And this is the comparison between glucose uptake high versus those glucose uptake intermediate or medium. And as you see, the most prominent changes that are highly significant in the statistics, and then also especially more so between the glucose uptake high versus the glucose uptake low, and as between the glucose uptake high and intermediate. And and as you know, then these gene sets that are highly upregulated for the cell cycle arrest, for the inflammatory response, especially the so-called senescence-associated secretory pattern, as well as the upregulation of the pro-survival factors. These are all characteristics for the senescent population of somatic stem cells. And indeed, we have also compared this with the senescent signature provided by this group of authors that summarize the senescent signature from the mouse model from 16 different studies and meta-analysis. And then we see a very high score for all of these upregulated signatures for this glucose uptake high. That means then the glucose uptake high is almost equal, and if not identical, to the senescent cells in the human hematopoietic stem cell compartment as we age. So we have also further on, we have performed this gene set enrichment analysis of all these single cell transcriptome data. So the comparison was, first of all, between glucose uptake high versus glucose uptake intermediate and low. And as a reference, we have also, as I mentioned, compared also the CD34 positive cells, the whole population from patients older than 60 versus then from healthy subjects older than 60 versus healthy subjects that are younger than 30. And we see, surprisingly, a very similar pattern in this gene set analysis for metabolism of amino acids, for the P50 pathway, which is also very characteristic for the senescent cells, and the mitochondrial complex and oxidative phosphorylation, etc. And also in the downregulated gene sets, so you see similar patterns of change. So altogether, I hope I have been able to convince you that this glucose uptake high subset, this few percentage of cells in the hematopoietic stem cell compartment in elderly is equal to the senescent population. And as you know from the literature, that the trend nowadays is that we can target those senescent cells, then to rejuvenate the stem cell compartment. In murine models have shown that secretion of the inflammatory cytokines by the senescent population, this secretion is actually responsible for the suppression of healthy hematopoietic stem cells in mice, and especially those healthy hematopoietic stem cells with the young phenotype. And in animal models, they also have made use of this upregulation, enormous upregulation of all the pro-survival factors of the PCL2 family, and inhibition of these anti-apoptotic factors or pro-survival factors, like with venetoclax, which is an approved drug, is able to eliminate all the senescent or part of the senescent hematopoietic stem cells in mice, and allowing the healthy hematopoietic stem cells with young phenotype to thrive, and resulting ultimately to rejuvenation of the whole bone marrow. And not only that, this study from Baker, or this study actually from Cho, from the group of Cho in Gainesville, have shown that not only are the hematopoietic stem cells rejuvenated in getting rid of the senescent cells, but also the stem cells for the mesenchymal stromal cells, the stem cells for the bone marrow niche, they were able to be rejuvenated. So therefore, our conclusion is that the glucose uptake high subset in aging human bone marrow is highly enriched for senescent cells, as shown by our series of studies, first of all with transcriptomics, and then later on with a transcriptomic with single cell RNA sequencing, and with the gene set enrichment analysis at multiple layers. And we have provided unequivocal evidence that this subset is enriched for stem cells, for senescent cells, and the percentage of glucose uptake high subset in hematopoietic stem cells increases continuously with age. And this metabolic property can be exploited to isolate senescent cells. And as you know, nowadays we still have no good marker or marker constellation for enrichment of senescent cells. And this method that yields very viable cells, glucose uptake high subset, might be the answer for many questions that we can use this method to enrich the senescent cells and to trace them, and also to follow the mechanism, how senescence develop in the human bone marrow. And moreover, as these senescent cells are addicted to survival factors and to elevate the glucose metabolism for survival. And in animal studies, inhibition of anti-apoptotic factors has led to depletion and reduction of senescent cells, leading ultimately to rejuvenation, not only of the bone marrow, but also of muscles and also the cardiovascular system. So in analogy, inhibition of glycolytic pathways may have similar effect on rejuvenation of human hematopoietic stem cell compartment, as well as other somatic stem cell compartments. So thank you very much for your kind attention, and I would be pleased to entertain any questions that you have. So thanks.
Thank you very much, Dr. Anthony Ho,